Our next speaker, Dr. Bill Andrews. Again, telomeres are the key, we think, to life. And nobody knows more about telomeres than Dr. Bill Andrews, so I'll invite you up on stage here, Bill. He's been working on telomeres for a long time, how to keep them long. And if you can keep your telomeres long and keep your cells healthy, then maybe you can keep yourself around for a long time and healthy. So, Dr. Bill Andrews. There you go.
Thank you. Is my mic on? Okay. I just want to qualify. My focus is aging. It's just that right now I think the priority is to work on telomeres to solve the aging process. And now, so a lot of people, I want to begin with clarifying a few things. A lot of people think that I'm obsessed with curing aging because I'm afraid to die. And that's not true. This is a picture of Star Trek, First Contact, when they make first contact with alien life. This is a big thing that's going to happen one day from now, and I want to be there when that happens. Okay? It's not a fear of dying. Okay? I just don't want to miss out. I'm fear of missing out. Okay, another thing that I want to clarify is that there's a lot of things out there now that people are saying reverses your aging. And this is because of biomarkers of aging. And I want to make it clear that just because you reverse a biomarker of aging doesn't mean you reversed aging. Is there anybody here who does not know which photo of Betty White was taken first? This is the biomarker. When a patient comes into your office and says, "Oh, I wish you'd make me young again," this is what they want. That's aging. Okay, aging is whatever's going to. We could take the wrinkles away on the photograph on the right. We could change the hair color, but she would still look old, and she would also feel old and behave old. And I don't want to use the word old as much. I'd like to use the word longer lived instead, but I accidentally sometimes say old. Now, this is what is reversal of aging. And what I call, so this would be the biomarker. And that's what every one of your patients wants. They don't want to just see their DNA methylation patterns change or their telomeres get longer. They want to see this. Okay, I call this the Betty White test. And the sad thing is, no one has ever passed this test yet. And I believe that we can do this, okay? I've been studying aging my whole life, and I believe that we know enough about aging that we can do this, and I'll be talking later on, if I have time, as to why we haven't completed it yet. So my subject is curing aging. I talk on a lot of different things. I talk about what aging is, why we age, how we age, and how not to age. And I want to say there's a lot of theories on all four of these things. But what I want to talk about today is that there's actually facts, not theories, that explain everything. And we don't need to resort to the theories. I'm not saying the theories aren't true. They might be also playing a role. But we will age despite any of the theories, just based on some facts that we know that I'm going to discuss today. Now, I also will be talking, if I have time, on why aging isn't cured yet. Okay, since I'm saying we know so much about it, why haven't we cured it yet? And also, why do people think that it is cured? But today, because of lack of time, I'm going to be just focusing mostly on how we age. But I'm going to just spend a minute or two on everything else. I'm going to skip over like my typical background on telomere biology that I'll be talking about a little at the end. But you can go to my website at www.crsi.com and see lots of videos on all these subjects that I just mentioned. And also I'm going to skip talking about who I am and my track record, but because those can be found there too at the same website. So on with the talk. First thing is what aging is. What is aging? And bottom line is just decline and failure of multiple tissues and organs. Really, that's all it is. Okay, a lot of people talk about what aging is, but they'll talk actually about why and how, and those will be different subjects. But I'm not going to talk about this today. As I said, that's just the bottom line that I conclude in most of my talks, and you can, again, go to my website to get more on that. The next question that I'm not going to spend a lot of time on today is the question of why we age. And more importantly, why have we never evolved a way not to age? Well, the answer is that it increases diversity when we eliminate the longer lived. And the more diversity within a species, the more likely we are to survive a rapidly changing environment. So those species, so if you create a new species and you put it on a planet, guaranteed eventually that species is going to evolve an aging process. Just as likely as if you toss a coin 100 times, you're going to get a heads at least once. It's all statistics and probability and logic and common sense that concludes that aging is inevitable for the success of the survival of the species. And more on that is at sierrasci.com. Okay, so this is where I'm going to spend most of my time today is how do we age? And as we all know, it begins with wear and tear. But it's more complicated than that. There's a lot more involved. And what I'm going to do is I'm going to describe a very simple example of tissues and organs. Wear and tear is the first thing that happens. But think of this brown or orange or whatever color it looks to you as an organ or tissue. And organs and tissues have a lot of stuff in common between them all. And one of the things they have is they have what I call, let's see if this doesn't always work, frontline cells. Frontline cells are like in a battle, the soldiers in the front line. These are the cells where all the action is, and they're the ones that are going to experience most of the normal wear and tear from normal operations. It's the kind of operations you have to stay alive, and these cells have a wear and tear rate, but they can only survive on average about a year. So just because of the normal operations of wear and tear. So here you have a worn and torn cell, and one of these front lines is worn and torn, and it gets lost. But that's okay when you're young or shorter lived because you do have other cells, reserve cells and stem cells, that can divide to replace these worn and torn cells, as shown here. And they also differentiate into those cells. And then there's a process of moving around in cell division that actually restores everything back to normal. So when you think about this, if this is really happening, should we age? Shouldn't this actually prevent us from aging if we can always replace the worn and torn cells? Now there's going to be exceptions with DNA damage and things like that, but in general, we should be able to survive a lot longer by continually being able to just replace these worn and torn cells. But the real problem is that our cells, humans at least, have a limited number of cell divisions. Their cells can only divide a limited number of times, and a newborn baby, that number is about 50. And, excuse me. So now if we go back to the beginning of a fertilized egg, that fertilized egg can divide 100 times. Now that doesn't sound like much, but if you do the math, you'll find out that a single cell dividing 100 times will create a mass the size of Jupiter. Okay, so that's plenty of cell divisions for us to be able to survive. 2 stages, at one cell divide, you have 99 left, 4, 98, 8 cells, you have 97 divisions left, 96 divisions left. Newborn baby, as I said, has 50, and then it gets shorter and shorter and shorter as you age. When you get into your 90s and over 100, you have less than 10 cell divisions left. This is called the Hayflick limit. It was discovered in 1961. Before then, people used to think that human cells could divide indefinitely, just like bacteria can in a petri dish. So when the cells start dividing, have reached their limit on number of times they can divide, they become senescent. And so this is showing a few senescent cells, and that senescence actually starts causing decline in your tissues and organs. And when you get a lot of senescent cells, it gets even worse. But here's now the real problem. You still have the cells, these senescent cells are still alive, they're still performing functions, and they still experience the wear and tear if they're a frontline cell. Now that frontline cell gets lost, and there's no cell to replace it because the cells are senescent. They can no longer divide. This now is not just tissue organ decline, it's tissue organ failure. So like things like chronic kidney disease and stuff, and I'm going to have a slide on that in a minute, is an example of where what happens there. Okay, so the consequences, I think that's the next slide. There's a lot of different diseases: liver cirrhosis, chronic kidney disease, dementia. All these things that I'm listing here have a connection to the Hayflick limit. Okay, now there's things that we can do to accelerate the rate that cells divide, or the rate that wear and tear occurs in our frontline cells. And we know a lot of high blood pressure does this, diabetes, alcohol, bad genetics. So if you have risk alleles that make your, like for instance, muscular dystrophy is a good example. You have the dystrophin gene, which actually causes your muscle cells to die faster. It doesn't affect you as a child very much at all. But because of the increased cell division, this is causing the number of cell divisions remaining to be less, and so you reach your 100 division level when you're still young, and you end up dying from muscular dystrophy. Inflammation, oxidative stress, pathogens, injuries, and air quality. Okay, now most of the hallmarks of aging are also related to wear and tear. Okay, and so I'm going to just go through those really fast. I've got 8 minutes left. And decreasing wear and tear is, I should have put this slide later on when I talk a little bit about how not to age, but there's a lot of things we can do to decrease wear and tear. Antioxidants, anti-inflammatories, on and on and on. Vitamin D is one of my favorites, sunscreen. But the question that I really want to ask is, what's more important? Figuring out wear and tear, how to solve the wear and tear problems, which there's 100,000 products already out there to do that, or solve the limited cell division problem. And there's practically nothing out there right now. There's a few products that make claims to do it, but nothing actually prevents the limited cell division. Maybe it extends it a few to 102 or 103, but nothing actually prevents that. Well, in truth, we have to solve both, okay? Because I don't want to say that even though I'm talking about a fact, I don't want to say the theories aren't true. So we have to work on everything. But what if we did solve the telomere, the limited cell division problem? That would make wear and tear a lot less relevant because of the fact that we can always divide cells to replace those damaged or lost cells. And so the question comes to mind, why is human cell division limited? And the answer is telomere shortening. We showed that back in the early 1990s when I was at Geron Corporation. It was definitely the thing that prevented, if we could keep telomeres long, it would prevent senescence. So in case there's somebody here who doesn't know what's a telomere, I'm going to quickly go through this. If we zoom in on a human body, we see that a human's made of 100 trillion cells, counting the bacteria and stuff like that, but mostly about at least 33 million human cells. Zoom in even further, we find every cell contains a nucleus, which you all know. Zoom in further, we see the chromosomes. And the telomeres are like the caps of your shoelaces, okay? They're on the very tips of your chromosomes. They actually protect your chromosomes like the caps on your shoelaces do. It's just that the caps on your shoelaces get shorter through wear and tear. telomeres don't actually get shortened by wear and tear as much. They can if you have a bad lifestyle or bad genetics, but it's mostly other things which I'll mention in a minute. But I want to zoom in on a telomere. Telomere, when you're first conceived, has a length of about 15,000 bases. Bases are units of DNA measurement. But as soon as your cells divide, every single time they divide, they get a little shorter. And they don't, so a newborn embryo has 15,000, they get shorter and shorter as your cells start dividing, not from wear and tear. Every time your cell divides and everything's duplicated inside that cell, the new daughter, to make the two daughter cells, everything has to be duplicated, and one of those things is the chromosomes. And when the new chromosome is made, it's made with shorter DNA. Okay, there's a lot I do talk about on other videos as to why that is. But as a result, our telomeres, by the time we're born, our telomeres have already shortened from 15,000 bases down to 10,000 bases. And it doesn't stop there because we have a lot of cell division yet to go. We've got to grow, heal wounds, fight infections, do all kinds of things that require cell division. And our telomeres get shorter and shorter. And when they get down to 5,000 bases, our cells go into senescence, our cells lose the ability to function, and we die of old age, or being longer lived. Let me go over that again. We're conceived at 15,000 bases, we're born at 10,000 bases, and we die of living too long by the time our telomeres get down to 5,000 bases. This is an actual ticking clock. This is not a theory. Okay, every person who grows human cells in petri dishes, in the lab, and all over the world knows that the number of cell divisions they have is limited by the telomere length. Okay, so they have to constantly replacing their cells with cells that have longer telomeres. I like to think of these telomeres as ride tickets at an amusement park. You get a bunch of tickets when you go to an amusement park, you ride a ride, you lose a ticket, and then you go on another ride, you lose another ticket. Because one of the things that I study a lot is the functions inside of the cells, the biochemistry, everything. So I understand the mechanism of a cell, and I've never been able to figure out how could a cell know how many times it has divided and how many more times it can divide. And the only analogy that I could ever think of that a cell has the ability to do is have something equivalent to ride tickets at an amusement park. And I was glad when I first heard about telomeres because that was exactly what the ride tickets at an amusement park suggested. Okay, so more on how we age at www.sierrasci.com. I want to spend a few minutes on how not to age, and that begins with the discovery that telomeres don't shorten in our reproductive cells, our primordial germ cells. And that's, if they did, our children would be born with shorter telomeres than we have, and their children would be born with shorter telomeres than they have, and we would have been extinct a long time ago. And so I was brought into the field in the early 1990s to figure out why, and my team discovered the enzyme human telomerase. This is a cartoon showing the factory-looking thing is telomerase bound to the end of a squiggly green DNA there, and what it's doing is lengthening the telomeres. So this only is true in our reproductive cells. We are doing two different approaches to try to figure out how to get this process to work in all of our cells. One of them is called gene induction, where we're creating like that little green thing that just dropped down is like nutraceuticals or pharmaceuticals that will bind to a repressor, dislodge the repressor from the gene that shuts the gene off. Since our reproductive cells produce telomerase, that means we all have the gene in all of our cells. So we're just trying to turn that gene on. That's one thing we're doing. Another thing is gene therapy, where we are actually think of it as soap bubbles. The gene therapy in the human cells, a soap bubble. The gene is inside, the telomerase gene is inside the smaller soap bubble, and they fuse, and the gene gets in there and produces lots of telomerase. So that's what we're doing. That we're doing at Sierra Sciences. More on that there. Let me just say, the bottom line is there's probably a lot of things that cause aging, but no matter what else we do to cure aging, our lifespans and health spans will still be limited to at maximum 125 years if we had the perfect genetics and perfect lifestyle. And unless we do something to solve the telomere shortening problem, we'll never exceed that. Okay, so I'm going to skip the why is aging not cured yet, but it mostly has to do with the systems that we have. And I'm going to skip the why do people think it's real. But those are on my website. I just want to say living is the greatest thing that ever happened to us. I love living. I don't want to see living become any less lovable for any of us just because of this dumb thing called aging. And let's all be there. Thank you.